So this is the first sperm acrosomal matrix protein to have a crystal obtained and the crystal structure resolved.
Both SLIP1 and SASON-V are found to be gamete-specific protein. So these proteins are uniquely expressed only in the SLIP1 in the sperm and testis, and SASON-V in the egg and in the ovary. We wanted to analyze the crystal structure of the SLIP1 molecule so that we can understand how this molecule is integrating on the egg membrane surface using SASON-V as a target.
Author's lab cover this SLIP1 protein. The protein, to me, I do the work to crystallize it and determine the crystal structure.
We found out that this molecule forms microfilaments structures to anchor to the egg surface prior to the fertilization process for the binding.
It's a pretty interesting biological structure, and it opens these questions. Is this structure normally forming in the acrosome? Is it forming at some stage during the fertilization cascade? And are those filaments involved in binding with the vestments of the egg?
The spiky filament we have observed is actually the result of analysis of the crystal structure. So it is not obvious from looking into the crystal structure itself. It is after analysis of how all these molecules arrange in the crystal. We see that they arrange in such a special way that they form a helix, a long range.
In our lab, we're looking at the cellular level and the subcellular level, what's inside an organelle and what's on the surface of a membrane. He can go down to the molecular level and understand where atoms are positioned in space.
We are looking into an atomic model, or maybe a model with multiple molecules, to characterize which residue is important for the biological significance of interactions.
It's just a fundamental structural finding like this opens all kinds of new hypotheses in thinking about the fine molecular nature of the fertilization process.